Okay, good morning. Thanks to the organizing committee for inviting me and giving me a chance to talk a bit about our data on Tetherin and direct cell-cell spread of HIV. Around 2 years ago, Tetherin was described as a cell host restriction factor and is induced by interferon alpha and part of the innate immune response against infections, and therefore is part of the antiviral state in cells. Tetherin literally tethers viruses or budding viruses to the cell surface and thereby inhibits the release of the virus. In HIV infections, the viral protein VPU antagonizes this effect by down-modulating it from the cellular surface. In HIV-2 infections, envelope does the job, as does NEF in SIV infections. On the cell surface, Tetherin is actually localized in lipid raft-rich membranes, microremains, which during the course of the viral life cycles actually focus the virus assembly, the virus budding, as well as virus entry. Further, lipid rafts are also involved in direct cell-to-cell spread of viruses through the virological synapses. Therefore, Tetherin is well located and well situated to have an effect on direct cell-cell spread of HIV. When we talk about cell-to-cell dissemination, we talk about the direct spread of a virus from an infected effector cell through the virological synapses into a target cell. We distinguish between viral transfer, where virus is passed from an infected cell directly to the target cell, and viral transmission, which comprises the viral transfer plus the productive infection of the target cell. Direct cell-cell spread is by several orders of magnitude more efficient than cell-free spread, and also exhibits way higher replication kinetics. It is considered to be the predominant mode of HIV spread in cell cultures, as well as secondary lymphoid tissue. It is actually on debate as to whether direct cell-cell spread might be a viral escape strategy from the immune system or from drugs. In our study, we used a human T-cell line, SubT1s. These are stably transduced with a human Tetherin gene. Here you could consider Tetherin-positive cells, outlined with in red, or with an empty vector, considered Tetherin-negative cells. We used the clonal reporter virus in its wild-type form, or the Delta VPU variant, in which we introduced double stop codon in the beginning of the coding region of VPU. We first, on the left side, confirmed the absence of VPU expression from our Delta VPU virus, and then in the middle, assessed the effect of VPU on Tetherin down modulation by flow cytometry. The effect we've seen by VPU is similar to the effect seen by other groups during the last 2 years. We used, as I said, the replication-competent reporter virus, which expresses eGFP from an iris downstream to NEF. In the viral genome, VPU and viral envelope are arranged in an overlapping fashion. So I don't know why that happened, but there should be a Western blot on the side showing viral envelope and viral P24 on the same levels. So we don't see an effect of introducing VPU stop codons on envelope expression. We then infected Tetherin-positive and Tetherin-negative cells with the wild type and the Delta VPU variant, and assessed the GFP expression profile over the first 48 hours of infection. We didn't see any differences regarding Tetherin and VPU during any of these initial time points. Therefore, we can say that VPU and Tetherin do not affect the initial cell-free replication or infection kinetics. To investigate the role of Tetherin in direct cell-to-cell spread, we set up a co-culture system in which we've been able to assess viral transfer and viral transmission from the same infections. We did so by co-culturing infected effector cells with pre-stained target cells and assessed transfer 6 hours later and viral transmission 24 hours later by flow cytometry. We detected transfer by intracellular staining for P24 and transmission by viral-derived eGFP expression. We set up the system, as you see in the top, in a way that cells were permitted direct contact, and in a control way on the bottom, where cells, infected cells and uninfected cells, were separated by a virus-permeable membrane. We didn't see any transfer or transmission events in the control setup at their respective time points. So what we found is that when Tetherin is expressed on the surface of the effector cell, the transfer of the delta VPU virus is restricted. However, we don't see a restriction of the wild-type virus by Tetherin. We then decided to look into more detail into the interrelationship between Tetherin and VPU in this matter, and we found that when the effector cell expresses Tetherin, that the wild-type virus is transferred more efficiently to the target cell than the delta VPU virus. That's on the left. And interestingly, on the right side, we see that in a setting where the effector cell does not express Tetherin, the delta VPU virus is transferred more efficiently. When looking at viral transmission by the means of detecting EGFP from the virus, we've seen a very similar pattern. Therefore, I think we can say that Tetherin restricts cell-to-cell viral transfer and transmission, that this restriction is overcome by VPU, and further, that the delta VPU virus has an advantage in transfer and transmission when there's no Tetherin around. We then wanted to look on Tetherin expression on the target cell, so the cell where the virus spreads into. Generally, the virus spreads more efficiently to Tetherin-positive cells. And again, we've seen the same pattern with viral transfer and viral transmission. We then wanted to investigate whether Tetherin has an effect on the infectivity of the transferred virus. We assessed the infectivity of the transferred virus by dividing the percentage of infected target cells of this batch by the percentage of the target cells which has virus transferred to early on. We then put the delta VPU and the wild-type variant into relation and called it an infectivity ratio. When this ratio was equal to 1 or around 1, we can say that the wild-type and the delta VPU virus are equally infectious. When it's above 1, the delta VPU virus seems to be more infectious, and when it's below 1, the wild-type virus is more infectious. Generally, we can say that these ratios did not significantly differ from 1. However, when looking at the Tetherin expression profile of the effector cell, well, if you compare the blue ones to each other and the red ones, you can't see a difference. However, when we're looking at the target cell, then we've seen, again, blue ones to each other and red ones to each other, that there was an increase of the ratio by around 45%, indicating that the delta VPU virus has an advantage in here. I would like to say that there's a couple of groups working on the same questions at the moment. There's a group of Olivier Schwartz in Paris and a collaboration of Claire Joly and Stuart Neal. Both groups presented during Cold Spring Harbor this year, and the group of Olivier Schwartz recently published a few weeks ago. So what we found in our group was that Tetherin actually restricts viral transfer and transmission, and that this restriction is overcome by VPU. We've seen that VPU actually might present a fitness cost to cell, specifically in cell-to-cell spread. However, this fitness cost is outweighed by the VPU advantage of the anti-Tetherin action. We found that the infectivity of the transferred virus is not affected by Tetherin, and we suggest that Tetherin might be actually involved in the virological synapses, and there not only in the effector cell, but also as part of the target cell. I would like to thank my supervisor, Dr. Mark Wainberg, Dr. Chen Liang, who contributed cell lines, as well as Richard, Tamara, Aaron, and members of the lab, the sponsoring agency, and I would like to acknowledge the people who contributed reagents through the NIH. Thank you very much.
So, is there a question from the audience?
Just wondering, do you see similar results in your sub-T1 cells when you induce Tetherin expression with interferon?
We haven't checked the effect by interferon, but we use this system because we can specifically take out the Tetherin part of the interferon response. And while interferon stimulates quite a few things, and by this method we saw we can do specified. To answer your question, we didn't do the same with interferons because we could thereby differentiate between the target and the effector cell.
And do you suspect that you'd find similar results if you use primary cells?
I suspect so. The other groups I mentioned, they're working partly with transfected 292 T cells and then as, with primary cells as target cells, and they've seen similar effects. However, the role of Tetherin in primary cells is still on debate, and we don't know exactly how big the impact of Tetherin actually is in vivo.
More questions? No? So, Bjorn, did you consider testing specific inhibition of VPU and examining whether this inhibition has an impact on Tetherin-mediated restriction of cell-to-cell transfer and transmission?
Sorry again, if we...
Did you consider using specific inhibition of VPU in your system and to see if this inhibition has an impact on Tetherin-mediated?
Yes. I would like to test a specific VPU inhibitor if someone happens to have one. I think there's one group or one company who's said they have one. It's called BIT225, but I haven't got my hands on these yet. So the only means of what I did so far is just doing stop codons in VPU. I would like to test that, obviously.
Okay.